Protein synthesis is a complicated and vital process that constantly occurs in your body. Through this video, you will gain the information needed to be able to understand how protein is produced in your body. Protein is created through protein synthesis, a process in which DNA is first transcribed into RNA, which is then translated into protein.
DNA consists of two molecules of nucleotides arranged in a double helix ladder. Nucleotides are made up of one phosphate group, one deoxyribose sugar, and one nitrogenous base. There are four different bases in DNA: thymine, adenine, guanine, and cytosine. In the latter model, phosphate and sugar form the backbone of DNA molecules, while the bases form the rungs. Adenine and thymine are always bonded together, and guanine and cytosine are always bonded together. The bases are bonded with hydrogen bonds, where A and T have two bonds and G and C have three. This latter is then twisted into an anti-parallel double helix, as shown in the diagram.
Transcription is the process in which an RNA polymerase unzips a section of DNA. In this process, transcription factors, proteins, bind to the promoter region of the DNA. After it has done this, the promoter initiates the process where the RNA polymerase speeds down a DNA strand and copies it to make a segment of mRNA. There are several important differences between DNA and mRNA. DNA has a double strand, while mRNA has a single strand and is much shorter than DNA. Because it is smaller, the mRNA can move outside of the nucleus to go to the cytoplasm and find a ribosome. Lastly, the thymine in DNA is replaced with uracil, another nitrogen base, in mRNA, and mRNA contains ribose sugar instead of deoxyribose sugar.
Most mRNAs aren't ready to be translated to protein directly after being transcribed from DNA. They first must go through RNA editing. The original transcript is called pre-mRNA, and a cap is added to one side and a poly-A tail is added to the other side of the pre-mRNA. Pre-mRNA contains transcripts of both introns and exons, so through editing, the introns are removed by splicing to produce the final mRNA. After processing, the mRNA is ready to leave the nucleus.
Translation is the RNA-directed synthesis of a polypeptide. A polypeptide is an amino acid strand. Translation requires mRNA, ribosomes made of ribosomal RNA, transfer RNA, also known as tRNA, and codons. A codon is a 3-base code for the production of a specific amino acid. There is one start codon that is at the beginning of every protein. 3 of the 64 codons are stop codons. They are found at the end of every gene and mark the end of the protein. Transfer RNA consists of a single RNA strand that is only about 80 nucleotides long. Each tRNA carries a specific amino acid on one end and has an anticodon, a codon with the opposite bases, on the other end. A special group of enzymes pairs up with the proper tRNA molecules with their corresponding amino acids. tRNA's purpose is to bring the correct amino acids to the ribosomes.
There are 4 different stages that create a final protein structure. First, there is a sequence of a chain of amino acids. This stage is called the primary protein structure. In the secondary protein structure, the hydrogen bonding of the peptide backbone causes amino acids to fold into a repeating protein, in which the alpha helices and pleated sheets are not yet folded onto each other. The tertiary protein structure occurs through 3-dimensional folding of the protein due to side chain reactions. The quaternary protein structure is the final structure where there is protein consisting of more than one amino acid chain. This process is vital for your body. Protein is an important part of every cell in your body. Protein is also used to build and repair tissues, and it creates enzymes, hormones, and many other chemicals found inside of you. Furthermore, protein is a monomer of many very important parts of your body, such as bones, cartilage, muscles, skin, and blood. Because of this, your body would stop functioning if it failed to synthesize protein.